Hi, my name is Paula, and today I'm going to present you my work named Identification of Genes with Biocontrol Activity and Comparative Genomics between Isolated JAB1 and Related Species. The white mold. The white mold is a disease caused by the fungus Sclerotinia sclerotiorum that affects more than 600 species like soybean, tomato, potato, bean, sunflower, and others. The fungus have a resistance structure named sclerotia that is capable of resisting soil for many years, making it difficult to control the disease. In preliminary studies, the bacterial isolate JAB1 was capable of inhibit the mycelial growth and the mycelogenic germination of sclerotia in vitro bioassays. So the objective of this work was identify the genes related to the biocontrol activity. The whole genome sequencing was done with Oxford Nanopore and Illumina technologies. The trimming were performed with bow tie tool, nanofield, and PoreShop, and readings were assembled with using the Mazurka software. And annotation was done with Proca. To search for secondary metabolic gene clusters was used the software Anti-Smash, and in the comparative analysis of the genomes was used Orthovin2. 11 gene clusters were identified in our genome, including the antifungal compound fungicin, the siderophores petrobactin and bacillibactin, that are compounds able to make iron available. Thus, bacteria that produce these compounds may be plant growth promoters and potential biocontrol agents. Besides that, in another study, the presence of bacillibactin in culture broth had significant biocontrol activity against bacterial and fungal phytopathogens by direct antibiosis. And the antibiotic sericidin that in a previous study presented bactericidal activity against a broad spectrum of gram-positive pathogens and showed efficacy against multi-drug-resistant Staphylococcus aureus and others. So this compound might be useful to human health. And others, unknown compounds with no previous description. Therefore, they might be new bioactive molecules. In the comparative analysis with Bacillus anthrax cereus, subtilis, and bombiceticus, we found 48 unique genes in JAB1 genome, and among them is the set of genes related to the synthesis of sericidin. Therefore, JAB1 has the potential to inhibit Sclerotinia sclerotiorum and its resistance structure, as well as other pathogens and phytopathogens, and may present genes that encode for new bioactive compounds. Thank you for listening.